Okay, so today I'm going to be talking to you about a study that we did on autophagy using the ATG4 gene and the CSTB gene in Tetrahymena thermophila. To start with autophagy, autophagy is the process by which proteins and other cellular components are either recycled or degraded. This is brought on by a period of cell stress. The process of autophagy includes the formation of the phagophore, which ends up engulfing the proteins and cellular components into an autophagosome. The autophagosome then fuses with the lysosome, which, with the pH of the lysosome, then degrades the proteins and cellular components that were inside the vesicle. We use T. thermophila because they not only multiply quickly, but they also thrive at room temperature and are a simple organism to study. This is generally pretty important for the classroom setting that we are able to do our lab work in. We chose ATG4 because it produces a protein that is a member of the peptidase family. It's also involved in vesicle elongation. We also chose CSTB, which produces a cysteine protease inhibitor and is directly involved in the lysosomal stress response. To start off our procedure, we started with primer design. We selected genes based on various criteria and then used bioinformatics to create primers with them. We created primers based on GC content, melting temperature, and primer dimerization when accounting for the efficiency of the primers. We then did gDNA isolation using a D&EASY kit. We did this to create a store of gDNA, which was used for further experiments down the line. Our first gel electrophoresis was using the pink 1 gene and our selected primers. We did this first gel to determine if our primers worked properly, and we used the pink 1 gene as a positive control for that experiment. We then moved on to cDNA synthesis using a reverse transcriptase, which was used as another DNA store for further experiments. Our second PCR and gel electrophoresis was a comparison between log and starvation phases using our selected genes primers. We then analyzed these gel results based on their expression profiles. Our first gel was the log controls. So as you can see, there's no bands in lanes 1 through 4, which should be expected since this is a negative control. However, there's also no bands in lanes 5 and 6, which means that there is no ATG4 gene product in these lanes. This indicates to us that there is either no expression of ATG4 in this time period, or that the primers did not work properly. In lanes 7 and 8, you can see there's 2 faint bands here, which indicates to us that the CSTB gene does in fact is expressed during this time point. You can see in lanes 1 through 4 on row 2 here that there's also no bands. This indicates there's no ATG4 gene product in these lanes either, indicating that again, either there's no ATG4 expression at all, or that the primers did not work properly, as in the first row. You can see in lanes 5 through 8 on the second row that there's very clear bright bands. This indicates to us that the CSTB gene is definitely expressed and is expressed very heavily during the log phase. This experiment was comparing the 2 genes in different points of starvation. You can see here in row 1, all of the lanes have very clear and bright bands. You can see that these lanes are slightly thicker, indicating slightly more expression, but not notable. This indicates to us that the CSTB gene is definitely expressed during these points of starvation, which are for lanes 1 through 4, 6 hours of starvation, and lanes 5 through 8, 9 hours of starvation. For row 2 here, you can see that there is light bands all the way across here. This indicates to us that the ATG4 gene is expressed during these starvation periods, but not nearly as much as the CSTB gene. You can see that there's no real difference in the thickness of these bands. However, lanes 1 through 4 is starvation at 3 hours, and lanes 5 through 8 is starvation from 9 hours, or 6 hours. So overall, you can see that CSTB is expressed during the log phase and is expressed heavily during all phases of starvation 6 hours and 9 hours. This is generally expected since this gene is directly involved in the lysosomal stress response, and this response is generally pretty unique to autophagy and apoptosis, so it's expected to see the heavy expression. ATG4, however, is not expressed during log phase, at least according to the gel results, but is expressed during the starvation at 3 hours and 6 hours, but not as heavily as the CSTB gene. This is expected since the gene is involved with vesicle elongation. And because vesicle elongation isn't unique to specifically autophagy or apoptosis and is generally found throughout the cell cycle in different mechanisms, it's expected that it wouldn't be expressed especially during starvation and would be expressed in similar amounts throughout the cell cycle. As for future experiments, I would perform an experiment using the same starvation points for both genes. I wouldn't use the same shifted pattern we used in the first experiments in order to get a more accurate representation of how the two genes compare at the same time point of starvation. I would also do an experiment comparing the expression of those two genes with the pink 1 gene. Since we know that the pink 1 gene is definitely expressed during autophagy, we could use this as a positive control in order to be able to compare the two other genes during different time points, for example, log starvation, or just different points in their cell cycle to be able to see if it's truly expressed during autophagy. I would also perform an experiment during conjugation of T. thermophila using both genes. This experiment would be purely out of curiosity to see whether or not these two genes are expressed during conjugation. Since we could see that they're both generally expressed during starvation, it would be possible that these two genes would also be expressed during conjugation, especially ATG4, since it's involved in vesicle elongation and it's not unique to autophagy. I could see that it could possibly have an expression profile for conjugation. And here are my references. Thank you very much.